Hello, everyone. Welcome and thank you for dialing into a webinar today titled UltraBurst, Excipient Platform, Develop ODTs in a Flash with UltraBurst. My name is Andrew Bagwell. I'm the Marketing and Communications Manager here at SPI Pharma. Here at SPI Pharma, we help solve formulation problems using superior functional materials, and we really provide innovative solutions to you guys, global pharmaceutical and nutritional customers, to help you guys succeed. And I'm really excited to have with us Graham McLeod, who's going to be talking to you guys about our new UltraBurst Excipient platform. Before we kick off the webinar today, I wanted to give you guys a quick introduction of what we do here at SPI Pharma. We help solve all of your formulation needs with our large array of product lines, including excipients, antioxidant actives, drug delivery systems, paste mask actives, vaccine adjuvants, and pharma solutions. And we have a lot of experience in the industry here at SPI Pharma, over 75 plus years, where we supply the pharmaceutical industry with highly engineered products and innovative solutions. And specifically, during this webinar, we're really excited to talk to you about UltraBurst and how this innovative excipient platform for ODTs is a first-in-class platform with disintegration times under 10 seconds. And here to talk to you a little bit more about SPI Pharma and what we do, and about our innovative product UltraBurst, is my associate, Dr. Graham McLeod. Graham has his PhD in pharmaceutical technology from the University of Manchester, England, and has over 25 years of experience in the industry. And he specifically has a lot of experience in oral dose foam technologies and processes, as well as novel soft capsule technologies and drug formulations. And before I kick it off to Graham, I wanted to let you know that if you have any questions during the webinar, please type them out in the chat window below, and we will address them during our Q&A session at the end of the webinar. Also, in your console on the screen, you can view additional resources like access to relatable downloadable content and the ability to book a meeting with one of our associates or fill out the survey.
Hi there. Good afternoon, good morning, good evening to everybody. My name's Graham McLeod. I'm VP for R&D at SPI Pharma. And today it's my great pleasure to take you through a topic very close to my heart and to SPI's hearts in general. We're going to talk about the development of orally disintegrating tablets using a new novel excipient platform called UltraBurst, which enables us to achieve really fast flash-type disintegration times using conventional tableting technology. So before we start on the main topic of the webinar, I just want to introduce you to, for those who don't know, to SPI Pharma. We have a mission to make a difference in the lives of our patients, our customers, our employees, and the community we serve. We really are about providing solutions, innovative solutions for our customers and enabling us to solve their formulation challenges and problems. We have a global organization where we have presence in more than 50 countries. We have manufacturing in North America and in Europe, and we have our headquarters in Wilmington in the US. In general, we supply actives for antacids, excipients, drug delivery systems, including platforms for ODTs. We have face mask actives. We supply vaccine adjuvants. And we also have the capability to, by using our dedicated team of formulation scientists based in our lab in India, we can actually offer drug development services where we can take problems and work with customers to solve some of their specific challenges using our products and our expertise. Okay, so just to take you through today's agenda, obviously I'm going to introduce you to the UltraBurst platform technology, and then take you through how we went about developing the platform, and also looking at what the challenges are from a formulator's perspective when it comes to, in general terms, developing orally dispersible tablets, but also trying to always keep in mind the need of the patient. And sometimes there's a conflicting requirement there between what you want to do as a formulator and what the patient needs. And hopefully we can explain exactly what those challenges are and how to resolve some of those problems. And at the end of the webinar, I'll take you through a couple of case studies that we have with two different APIs using the UltraBurst technology. One is diphenhydramine, and the other one is loratadine. Okay, so what is UltraBurst then? In a nutshell, it's a completely novel, revolutionary system that enables rapid flash-like disintegration in tablets using conventional DC process. We believe it is best in class in terms of delivering fast disintegration times, maintaining tablet hardness and robustness, friability, low friability, and high API loading. As mentioned, it's designed for direct compression, so therefore costs to produce tablets are low, and we believe not only is it effective for low-dose small tablets, we also think it gives formulators opportunities to develop ODT-type products where in the past this wasn't possible. Okay, so let's take a look at what the key challenges are and targets are for developing an ODT. The first one is achieving the requisite disintegration time, and that would be 30 seconds if you're in the US following the US monograph, or 3 minutes in the EU, and that's regardless of the size of the tablet. Obviously, disintegration time being as low as possible is the key target for any ODT and any ODT platform. Another consideration is tablet hardness, and when it comes to ODTs, there's no real set limit on hardness. It's really more to do with limiting friability. And similarly with friability, although friability is measured routinely for swallow tablets, a limit for ODTs is not always clear, and there's not a lot of work in the literature to guide formulators on what the best target should be. We try to get friability values as low as possible because obviously your ODT may be subject to a downstream process, for example, packaging and transportation. And even when it gets to the patient has to open the pack and get the tablet out, and hopefully the tablet remains intact when the patient does that. Other approaches to reducing friability with different platforms and different approaches can give issues with friability, and we believe UltraBurst is designed in such a way where we can kind of optimize the two conflicting requirements of fast DT and low friability. At the end of the day, any API that's incorporated into an ODT system, we have to look at dissolution and also potential for some APIs to potentially be absorbed from the oral cavity. Key underlying requirement for all these systems, because they dissolve and disintegrate in the mouth, is the palatability. So taste masking and a nice organoleptic experience for the patient is also critical. And for any given ODT, a process, preferably one that's simple and cheap, such as direct compression, is always an advantage. So as we discussed, achieving a fast, rapid ET, preferably less than 10 seconds, is a challenge, particularly when we're trying to get a lowly friable product. And that is easier to achieve when we have low dose and low tablet weight formulations. As we get to higher drug loadings, higher dose of APIs, larger tablets, which may have prolonged wetting times, the challenge becomes even more difficult. With higher doses of APIs, compression is harder. Some of the APIs we use can be difficult to compact, particularly if they are coated with taste-masking polymers. And if you want to keep those taste-masking polymers intact, you don't want to be compressing the compact to very high force, as this would then cause problems for the patient in terms of bitter or unpleasant tasting drugs. So always, whatever we do, we've got to focus on positive organoleptics for the patient. UltraBurst gives us the opportunity to develop much wider range of APIs at wider tablet weights and higher drug dosing than other conventional approaches. Let's take a look at some of the factors that influence the disintegration time. The main one of these is the wettability. This is the ability of your compact to imbibe aqueous media, whether that be water in vitro or whether it be saliva in vivo. Generally speaking, ODTs have a higher porosity, and that enables water to ingress into the system. And the ability of the system to wick water will affect how quickly the compact is wetted and in turn how quickly it will disintegrate. Higher amounts of aqueous media absorbed should give you faster swelling of the matrix, and that swelling should be associated with faster disintegration. However, you want this swelling to happen without any propensity for the compact to gel, as that can give prolonged disintegration time and even an unpleasant texture for the patient. By including some propulsive agents which can expel or increase the swelling and the breakup of the tablet, we can achieve these targets fairly efficiently. As well as the swelling and the expulsion of agents, the speed at which the compact softens is also important. And all these factors overall contribute to the overall organoleptic experience that the patient receives. And also by focusing on each of these, we can ensure that we optimize the overall quality attribute that's important, which is the disintegration time. We've talked a lot about achieving a rapid disintegration time, but what we should also consider is what method should we use to measure DT, and how close is that to what happens in vivo. Standard method, which is in the monograph, is really not that realistic. Relevance to what happens in vivo is questionable. There's lots of water and lots of agitation for dose form in that method. So really, it's not that realistic, and it's unlikely to give you a simulation of what happens in vivo. There has been a lot of work by various other scientists and formulators looking at adapting that method to make it more relevant, using dripping water onto tablets sitting on meshes, measuring force of swelling of tablets, etc. Looking at the wetting time or the dispersion time of a dose form. And those methods do appear to be much more close to what happens in reality when the patient takes dose form. We've also done some work with University of London School of Pharmacy on what's called an oral cavity simulator. And essentially that's just like a mechanical mouth, and it uses a fabricated system based on an engineered mouth that resembles what happens in vivo. And I'm going to talk a little bit about that in more detail later on in this presentation. As well as the method, what's really important as well is what media should be used in the method. Should we use simulated salivary fluid? Should we use water? Or should we use buffer? And essentially this depends on the individual project and the closeness of the method to the reality of the in vivo scenario. Because at the end of the day, what we're trying to do is we're trying to show some sort of correlation between what happens in the lab when we test for DT and what happens when the patient takes the tablet. So what sort of disintegration time do we get with UltraBurst? You can see in this table here a range of different tablet weights, all on the low side in terms of typical target weights of between 50 and 200 milligrams. And therefore, regardless of the hardness and the compression force used to attain the tablet, you can see that disintegration times of less than 10 seconds is easily achieved, particularly below 150 milligrams. And for tablets as small as 50 milligrams, we're getting really, really low disintegration times of 3 seconds. You also should note that the disintegration time essentially is proportional to the weight of the tablets, going from 3 seconds at 50 milligrams up to around 9 seconds at 200 milligrams. And the reason for this is really, you can see in the picture on the right-hand side, where you can see that the mechanism for the disintegration of the tablet is one in which is consistent. So as we increase tablet weight, we have the same mechanism of disintegration using a expulsion of the components based on an effervescent component of the system, and that remains consistent across the different tablet weights. And that enables us to achieve really impressive DTs, less than 10 seconds, for tablets that are as high as 200 milligrams in weight. So as I said previously, not only does UltraBurst enable us to get very rapid disintegration times for smaller tablets, it also means that for larger tablets, we can start thinking about a ODT type system for products where the formulator in the past would not have been able to formulate as an ODT. You can see right here in the table on the slide, we're looking at 500 milligram tablet weights of 2 different APIs, and we're comparing the disintegration time, the friability, and the hardness of UltraBurst-based tablets versus alternative platforms. The main thing to pick up here is that, first of all, UltraBurst gives you the shortest disintegration time, just over 10 seconds, and it compares extremely favourable to the other platforms that we tested. And in some cases, we actually found that those platforms gave you product that didn't seem to disintegrate at all. So although these systems are formulated for rapid DTs in mind, they're not all the same. So not all these systems are equal. The other thing that we can pick up from this bit of work is the differences between the APIs. So in the top table, we're looking at a 10% loading of a micronized API. In the bottom table, this is data for our SPI in-house face mask APAP, which has a hydrophilic type coating. And this really demonstrates that depending on the properties of the API, whether it be particle size, surface activity, wetting capability, etc., you can get widely differing results from different systems. And you should always bear that in mind when you're looking at developing an ODT. So criteria like particle size, surface properties, and drug loading are all hugely important when it comes to optimizing and developing the targeted product profile. The categorization of ODT systems is increasingly well researched in the literature. Typically, one of the things the researcher can look at is the so-called solid fraction versus whatever attribute we're interested in, for example, tensile strength. So in this approach, what we do is we make our tablets, we compress them at given compression force, and then we measure the tablet dimension. We also measure the true density of the blend. And from these data, we can then calculate what's known as a solid fraction. Solid fraction is essentially just the inverse of the porosity. So the higher the solid fraction, the less spaces there are in the tablet compact. And typically that would be a negative thing for an ODT, where you want high porosity, you want a structure whereby water can imbibe into the tablet readily and force disintegration of the tablet. If you look at the 3 different platforms here, what we're looking at there is essentially what we want is to try and have a linear relationship whereby as we increase the solid fraction, we increase the strength of the tablet. And you can see that that is the situation with UltraBurst, whereas especially with the system platform 2 here, what happens is that as we increase solid fraction, we're actually not improving the tensile strength or the robustness of the tablet. That means that we are hitting the tablet with a higher force, we're reducing the porosity of the tablet, but we're not getting any benefit of that in terms of robustness or friability of the tablet. The data in this table are all done using 10% loading of ibuprofen, and you can see that essentially UltraBurst is performing well, and it's showing this linear relationship, which means there's a nice, robust, strong compact being formed with UltraBurst, and it's superior in terms of its tensile properties compared to the other two platforms. So I talked earlier about sort of paradoxical requirements for an ODT, where you want as strong a tablet as possible, but you want as short a disintegration time as you can get. And here we've looked at the tensile strength of UltraBurst compared to different platforms and looked at the resultant disintegration time. And again, you can see that UltraBurst performs extremely well compared to the other two platforms, where we're getting essentially a pretty flat line of disintegration time, even as we increase the tensile strength to very high values. So typically your tensile strength target might be around 2 megapascals, but even if we increase beyond that, it doesn't significantly reduce disintegration time for the UltraBurst tablets. In terms of what is the target tensile strength for ODTs, this is a question that's kind of open to interpretation. We know that for small tablets, typically the range of tensile strength target is around 1.7 to 2 megapascals. For ODTs, it may be preferable to go slightly lower, but you can see UltraBurst here stands up well in terms of that target versus having a very low disintegration time. Remember, this is very important when it comes to downstream packaging, and there are ODTs in the marketplace which are very friable and which are not great from a patient perspective. When they get the pack, they open the pack and the tablet is broken or in several parts. Using UltraBurst, this phenomenon is less likely to happen, meaning we can be flexible in terms of target product profile and reduction of the friability of the product when it gets to the packaging process. So I mentioned earlier that there's many different ways to measure disintegration time, and that really the monograph apparatus is not really fit for purpose. And this is just some interesting work that I want to just review with the audience today around a study we did with developing an oral cavity simulator. So this is work run out of the University of London School of Pharmacy there. And essentially the idea here was, can we replicate using a mechanical system what happens in the patient's mouth? And we had an acrylic plate developed with what we call a silicon tongue, and that tongue can actually move up and down, so that can mimic the mastication of a tablet in a human's mouth. We can drip saliva onto that tablet, and then we can, using a camera, we can actually take photographs with time, and using those photographic images, we can actually measure the area of the tablet with time. And what you see is this graph on the right-hand side. So as water, as I described earlier, as water is imbibed into the tablet, that tablet will start to swell. You can see that in the area measurements on the graph. So in the early, between 0 and 40 seconds in this plot, the overall area of the tablets starts to increase. It then kind of plateaus around about 1 minute, and then as the system starts to disintegrate, obviously the area decreases, and that is shown in the measurements that are taken with the camera, and with time the total area reduces and reduces away to 0. So you can estimate the DT using this type of system, and you can compare it quite favourably to those results that you see in vivo. There are actually very few publications which look at the correlation between in vitro disintegration time and clinical or in vivo disintegration time. One publication which has been done by a group of workers in Japan is very interesting, in that they show a pretty good correlation between the in vitro disintegration times versus what they found in the clinical setting. What's really interesting about this on the whole is that these tablets are all generally low-dose APIs, probably quite small tablets, and they used a system called a tricop tester, which essentially was a design whereby saliva is dripped onto a tablet. That tablet sitting on a mesh system, and they could measure disintegration time by observation of the tablet as the saliva was dripped onto the tablets. So you see, if this wide range of different APIs, all fairly low dose, they actually got a range of in vitro disintegration times, typically in the range of between 20 and 30 seconds. And when they tested these products in the clinic, they actually found that the DTs were very similar. So there was this high degree of correlation between the value that they obtained in vitro and what they subsequently saw when they gave these tablets to patients. And this is really critical. We want to try to predict as best we can, the DT we see in the lab is what the patient experiences. So giving these results, which we just described, and you can see on the left-hand side there, this correlation that I described previously, where they have this kind of straight line relationship between the disintegration time in the clinic and what we found in the lab. What we thought would be interesting was to look at whether or not we get the same type of relationship with UltraBurst-based formulations. When we did these tests, we found, yes, there is a correlation, and it's pretty linear. And as the tablet weight increases from 50 to 100, 200 milligrams, obviously the in vitro DT time got longer, as I've shown you in the previous slide. What was also really interesting was that when we looked at the in vivo disintegration time, there was a very close correlation between those values that we'd seen in vitro and those values that we got in vivo. And what's really key for this technology is, overall, these in vivo disintegration times are all much faster than the ones you can see that are typically reported by the group in Japan, for example, where we have the in vivo disintegration time. You can see on this graph on the right is ranging between 4 and 11 or 12 seconds, whereas on the left-hand side, it's ranging between 18 to 35 seconds. So UltraBurst gives you this, first of all, gives you this correlation that's important, but also gives you this much faster disintegration time, particularly for tablets which are low dose in terms of API loading. So up until now, we've talked a lot about the challenges for the formulator in terms of achieving the rapid disintegration time. We've looked at what effect does tensile strength, friability, what effect does that have on DT? Can we circle the paradox when it comes to achieving low DTs and high tensile strengths? And we've seen that UltraBurst can achieve that. The other thing for any DC process, the other thing that the formulator has to really consider carefully is flowability and mixing uniformity of any blend or any formulation that's going to be used in the DC process. Here you can see, in terms of the product characteristics of UltraBurst, it has a low CARS index, low angle of repose, LOB is low, and this is inherent in the system in terms of we've tried to design it as best we can to be suitable for DC processes. Because as soon as you add API to the platform, whether that's micronized, sticky, low-dose API, or whether it's larger particle size, coated API, which is taste mask, one of the key challenges you're going to have as a formulator is the mixing, the blend uniformity, and the flow on the compression machine. So what I'd recommend formulators do in this situation is not just looking at the numbers, but also look at the tests as you perform in the lab. How does the material flow? What does the angle of repose look like? How does it compare as you play around with the different the formulation attributes and the different parameters that you have to play with in terms of excipients, platforms, etc. And then you've got to try to match as best you can those attributes of the platform, those requirements and the challenges that the API gives you, whether it's sticky, whether it's a low particle size, or whether it's the opposite, where it's a high particle size, which might be called disintegration. These challenges that I highlighted in the last slide are exemplified in this slide here. We're looking at the difference between two different APIs, loperamide in one case and loratadine in the other. Both are low-dose APIs formulated into small tablets, in this case 50 milligrams. The blend particle size, P50, is about the same. ODTs are about the same. And you can see here, not surprisingly, there's no challenges here in terms of hardness or friability, because overall the API is present at a low level. However, what was different between the two systems was the content uniformity. Not surprisingly, when you have such low-dose APIs for a DC process, depending on the properties of the API, you can get widely varying results from a blend and tablet content uniformity perspective. You can see here some of the reasons for that. Clearly, if you look at loperamide API, there's needle-like morphology, and that means that it's very difficult to get uniform blend using that type of material. On the other side, the loratadine is much more uniform, much more spherical API. Has some challenges in that it's sticky, but overall the results for content uniformity were pretty much as targeted. So all we had to do with the loperamide was either build in a processing step to improve content uniformity prior to direct compression, or actually what we did do was we looked at a blending approach using a technique called trituration, where we essentially, by using a careful blending technique, we managed to overcome the content uniformity issue that we'd seen initially, and we got a much improved content uniformity of between 90 and 103 versus an initial content uniformity of loperamide, which was 77 and above. So I guess the learning here is obviously APIs are all different. You have to look at particle morphology, particle size, the cohesiveness of the resultant blend, and the interaction between the API and the platform or the excipients that you're using. Then you need to take a step back and look at what's the best way to achieve the content uniformity that I'm aiming for. Can I do it simply by direct compression, or do I need to look at more at how I blend, my blending times and my blending approach, or even in certain circumstances, do I need to do some upfront process changes such as co-milling, for example. And this issue is demonstrated again in this table here. So the first thing is that although ODT platforms are many, they're not all the same in terms of the performance and their flowability. You can see here UltraBurst compared to another platform, significant differences between the flowability of UltraBurst because it's been designed for DC processes and this other platform. And then if you look at the addition of 10% of a micronized API, what happens unsurprisingly is the values for the various parameters that we're looking at, such as angle repose, bulk density, tap density, GI, and Hausen ratio, they all get worse essentially. So when you're adding API to a blend, you're not going to improve the flowability or the potential for segregation within that blend. You're going to make it much worse. And with UltraBurst, because it's been designed with good flow and good processability in mind, the chances are you're more likely to be able to run this on a DC process compared to some other platforms. So then, to summarise the discussion for the last half hour or so, hopefully I've been able to show UltraBurst is really primarily designed for this rapid disintegration, sub 10 seconds, flash-like performance from a AC platform technology. We understand the compressibility of the UltraBurst platform. We've characterized it well, and we've seen that the tensile strength, although it increases linearly with time, it does not greatly affect the disintegration time when you look at UltraBurst. That is not the case for other platforms. We have, internally within SPI, we have a lot of knowledge of developing ODT systems and understanding what the key challenges would be for a robust direct compression process. And we bring that to the table when it comes to looking at a range of different APIs, so that depending on the API properties, the drug loading, the particle size, the cohesiveness of the API, we believe that we can find a solution using UltraBurst with your challenging API and come up with a DC formulation that will give you a much reduced DT, and that DT in vitro will also be seen when it comes to what the patient's experience is as well. We also have some case studies, which I'm going to talk about next, where we show that the final tablet formulation with some digital APIs meets all the requirements from a stability perspective. So we've taken you through some of the theory, some of the challenges that comes when it comes to developing ODTs and formulating specific projects and getting around different challenges. The next part of the presentation, we're going to look at a couple of case studies that we've done at SPI, where we've looked at developing two different OTC APIs using UltraBurst as a platform, and I'll take you through each of these in turn, starting first with diphenhydramine ODT 12.5 milligrams. Okay, so the objective of this work was to try to develop a diphenhydramine ODT which was taste masked, which would have a rapid disintegration time and would be, to some extent, an improvement on the existing diphenhydramine pediatric 12.5 milligram product that's on the market, which is a chewable tablet. For those who are not familiar with diphenhydramine, it's an antihistamine. It's fairly soluble in water, and one of the main challenges of developing this API into a palatable dose form is the taste masking aspect. As well as making sure that we have sufficient taste masking, what we embarked on in this project was to try to significantly reduce the size of the dose form so that we had a very patient-centric small tablet that, if necessary, could be taken as conventional ODT, but also we could drop that dose form into a spoonful of syrup or water, and that would enable that particular format to be dispersed quickly and taken easily by a child. Another advantage of the formulation of UltraBurst that we were aiming to develop was its very rapid disintegration time, with a target of less than 10 seconds. So the following slides I'll take you through how we get on in trying to meet some of these challenges. So for this project, we're really focused specifically on developing very simple ODT based on UltraBurst, focusing really on the physical and the chemical attributes of the tablet, knowing that for each individual customer who might be interested in a similar product concept, they would add their own required flavorings, sweeteners, etc. So we're really just looking at a very simple formulation based on UltraBurst with some diphenhydramine resonate as a means of ion exchange complexation to case mask the diphenhydramine, and some lubricant in the formulation to ensure the DC process ran well. So the case masking approach, as mentioned, was using ion exchange resin. It's a fairly straightforward and simple process where we activate the resin in water. We then adjust the pH to around 6.0 and stir the ion exchange resin with the API for around 1 hour. We then would filter that product and dry it and aim for an LD of less than 6%. In terms of manufacturing the actual ODT, again, very simple process. We take the drug resonate, which is manufactured the way I've just described. We would sieve that through a 30 mesh, which in microns is 595 micron sieve. And then the resulting product we just blend with the UltraBurst before lubricating with magnesium stearate and mixing that for 5 minutes. So here's on the slide showing some of the powder physicals of the blend that we use to tablet the UltraBurst formulation containing diphenhydramine. You can see that the actual blend properties themselves are all very favourable in terms of being able to achieve a uniform flow from a DC process. And because we used the resonate process, we can actually target a particle size of the drug resonate of around 100 micron, which matches well with the D50 of the UltraBurst material. When we measured the blend uniformity, we achieved very low RSD values of less than 2% versus a limit of 6%. And you can see the data here on the right-hand side. Blend uniformity is, and the assay values are also well within the limits, around 97%. So having achieved good blend uniformity, the next stage of the development was obviously to show that we could compress that material into a robust tablet that showed the requisite dissolution properties. So we made a small batch of tablets on a lab scale, development scale compression machine, batch size of 5,000 tablets for runtime of around 2 hours, and we had no issues in terms of the uniformity of the flow into the dye or the resultant dissolution. You can see here on the right hand side, comparative dissolution of each batch, very similar for the UltraBurst based formulation, and not surprisingly it's faster than the chewable tablet, which, let's bear in mind, was developed not as an ODT but as a chewable, but it gives us a good comparison in terms of how the UltraBurst formulation compares to what was already on the market. In terms of tablet content uniformity, you can see the AV values of each batch are well within the target limit of 15 at 4.3 and 2.9 respectively. So looking at the tablet physicals from the product that we described in the last slide, quite a noisy chart here, but essentially this is just showing for 10 tablets, 2 different batches, the typical range and hardness and disintegration time that we saw with this product on the left-hand side, and with the weight variation and thickness on the right-hand side. Essentially, no surprises here, with tablet hardnesses in the range of between about 33 and 38 newtons, and disintegration times, again, within the range of around 6 to 8 seconds. So very rapid disintegration times as we intended to achieve. And again, looking at the right-hand side, again, this is representing 10 tablets. You can see very reproducible in terms of the tablet weight versus the target of 100 milligrams, and thickness, minimal variability between the different tablets. So the tablets that we made were then put on a formal ICH stability testing program. They were packed into HTP bottles and stored at two conditions, 2560 and also 4075. We did measure the physical as well as the chemical attributes, and there's no issues with the physical attributes. And what's shown here in this graph is the assay with time, and also the impurity profile of the RS with time. You can see the assay remains consistent for 6 months at 4075, and the impurity level rises slightly from 0 month, 1 month time point through to 6 months, but is well within the limit of 3%. We actually have a whole range of stability data for UltraBurst formulas at different tablet weights, in different packaging configurations. So we should be able to answer questions relating to the stability of UltraBurst tablets over when stored at 4075 for 6 months, and at 2560 for one year, and it's ongoing, so we'll have 2 years data when that time point comes. Okay, to summarise then, we developed a previously unavailable diphenhydramine case mask, ODT 12.5 milligrams, intended for pediatric use. Case masking approach we used was using ion exchange resin, and that masked quite effectively the very bitter taste of diphenhydramine. We've shown this formulation was stable for up to 6 months at 4075 storage conditions, and you can see that we had a very patient-centric dose form compared to the currently existing marketed chewable tablet formulation. Our ODT based on UltraBurst is a tablet weight of 100 milligrams versus 600 milligrams commercial chewable product, and our DT was 7 seconds, which is much faster than the commercial product. You can see the pictures at the bottom here just showing the differences in terms of the overall size of the UltraBurst-based ODT versus marketed product. And we think this would be a significant advantage from a pediatric perspective, particularly as it gives flexibility around the number of these small tablets that the child can take. And if necessary, it would be fairly easy to add these ODTs, as mentioned previously, to spoon containing water or syrup, and these would disintegrate rapidly, disperse rapidly, and the child would therefore have a very palatable dose form to take. They also could be taken along with food substances such as jams or applesauce, etc. So overall, we basically met the objectives of the study, producing a highly patient-centric dose form compared to what existed on the market. So the second project case study that I want to describe to you is one where we've done work with another API, loratadine, 10 milligrams, and again we developed a UltraBurst-based ODT and compared that to a product that already exists on the market. So in the following slides, I'm again going to take you through what the objectives of this study were and how close we got to achieving those overall objectives using the UltraBurst technology. So we're looking to develop a loratadine 10 milligram ODT based on UltraBurst. The loratadine API is micronized, which gives us another challenging situation to overcome in terms of achieving good content uniformity using this micronized API. For those who are not familiar, loratadine is an antihistamine and is widely used in children as well as adults. There are several marketed products. The one which we compared to against here, the features are given on the right-hand side of the chart. And the main issue with the specific commercial product that we identified was this very high friability. Really quite unacceptable friability of around 7% of the product, which may cause problems for patients in terms of when they open the bottle of tablets or the blister pack, they would see the tablets were broken, and this could put them off taking their medication. We're also looking at reducing the disintegration time. We measured it around 16 seconds for the Marty product, and we were aiming to get below 10 seconds using UltraBurst. And along with the issues of friability, there is also a relatively large tablet weight of 280 milligrams, which again we're looking to target 100 milligrams for our UltraBurst-based ODT. So here on this table, on the top left-hand side of this slide, you can see the formulation where we have the API, 10 milligrams, and we have added in the UltraBurst platform along with some peppermint flavour and some sweetener, sucralose, and lubricant. So again, you can see an extremely simple blend, and when we look at the particle size of that blend, around 135 microns for a D50. The API itself, where atidine has a very low D50 of around 6 microns. So we reckoned that this would cause us some challenges when it came to blend uniformity, and we're looking to see if we could achieve good content uniformity, good mixing with UltraBurst, with this specifically challenging API, which is micronized and also tending to be quite sticky and cohesive, so quite difficult to formulate for a DC process. Okay, so looking first at the physical properties of the tablets we produced, made them to hardness of 25 newtons, very low friability of around 0.1%, and that BT is well below 10 seconds, at 7 seconds. Surprisingly, when we looked at the content uniformity, we were happy to see that despite the fact that the API was fairly cohesive and had a very small particle size, we actually achieved very reasonable content uniformity for the blend, an AV value around 2.6. We manufactured the tablets using direct compression process, a runtime of 2 hours and a batch size of 5,000 tablets. And you can see from the graph on the right that the dissolution of the UltraBurst ODT and the marketed product are very similar. And from our content uniformity values of the tablets, you can see again the AV value of 2.6 is well within the target, which is less than 15. So an interesting point that I never covered in the last slide was the fact that when we tried to further reduce the size of the tablet, we actually found the inherent stickiness of the API become more and more of a problem. So if you looked at trying to formulate a mini tablet with, for example, a mannitol-based system, it was extremely difficult. We couldn't actually achieve a process without getting significant sticking to the punches on mini tablets. But it would appear that the cohesiveness of the API with atidine is somewhat masked as you increase the tablet weight to around 100 milligrams, and as you increase slightly the compaction force. So this slide summarizes the overall findings from the study, and you can see that we achieved the objectives, the main one of which was to produce a robust tablet with a short disintegration time using the UltraBurst technology. We almost half the disintegration time from 16 seconds to 7 seconds. Viability was much more acceptable for the UltraBurst-based formulation, around 0.1%, despite the fact that the hardness was very similar to the level seen for the commercial product. And overall, the tablet weight of 100 milligrams, around a third of the tablet weight for the loratadine product that's available commercially. You can see at the bottom here on the left-hand side, our product is thinner and smaller than the marketed product. And again, we think this is a much more robust and patient-centric dose form that lends itself to use both pediatric indications, but also with adults. So then to summarise what I talked about today, hopefully I've taken you through some of the challenges that you may experience as a formulator when developing ODT tablets. I showed the importance of the characterization of the formulation and the compact using the various techniques that I described, looking at things like tensile strength targets, physical characterization of the blend, looking at flow and morphological considerations, particularly as it relates to the API. Remember to look at not just the data, but also visualize how these materials look under test conditions. I've taken you through the approaches that we undertook to develop UltraBurst as a platform, and some of the different ways in which you can actually measure disintegration time, and the importance of the correlation between in vitro and in vivo DT and different ways in which you can achieve that, and showing that UltraBurst has this good correlation but also has a much more rapid DT time in vivo compared to other platforms. I've exemplified using case studies or ways in which we can formulate challenging or difficult APIs using UltraBurst. So in summary, UltraBurst basically is a direct compression based technology, enables you to have a rapid DT or a flash-like system using conventional DC processes. That means you can significantly accelerate the development time and produce a patient-centric dose form that has advantages over those products that are already on the market, but also can open up new possibilities around developing improved products or products where API loadings may be higher or drug doses may be higher, and that will then open up the opportunity to actually develop ODT-like formulations and products that you couldn't previously do. We have, as mentioned, within SPI, we have a team of specialised formulators who are very experienced when it comes to developing formulations and can help you speed up the development of your product that's maybe in development. So please reach out to us and speak to our team, and hopefully we can get you a sample and you can begin using UltraBurst yourselves, so you don't need to take my word for it of how good a system it can be. Hopefully you've seen the innovation that we undertake at SPI, and today I've spoken in detail about UltraBurst, which is our ODT technology. In SPI though, we have many other products and services that you may be interested in. For example, we have antacid actives, we have functional drug delivery systems and excipient systems that give you superior organoleptic and superior processing advantages. We have products that are applicable in soft capsules, our Sobril Special plasticizer range, and can give you significant advantages when it comes to drying times and stability of soft gel capsules. We have a soft gel capsule machine that enables us to undertake prototype development and formulation development work hand in hand, helping customers to speed up their development using a dose form that's still fairly rare in the industry, and particularly difficult when it comes to having lab scale or pilot scale equipment. This is a service that we have that maybe you find interesting that you could work with us on. We also have a number of taste masking technologies. We have a couple of taste mask products, the ActiMask range is available, and we are able to look at specific challenges that you may have regarding developing a taste mask API, and we can look at the best way in which we can quickly develop a prototype formulation that will have mask taste of bitter or astringent APIs, so you may want to eliminate some of the obnoxious tastes and flavours that are associated with them. So similarly, a whole range of products. And I mentioned we have a drug development business of Pharma Solutions. Pharma Solutions, we have the ability to, if you like, on an a la carte basis, offer services and support for whatever your challenges may be when it comes to drug development. Hopefully, an oversight of both the specifics of UltraBurst, but also the types of products and services that SPI Pharma can offer you in the future. So hopefully you've enjoyed the content of today's webinar. On the screen here is my direct contact number, email address. If you have any questions that come out of this webinar or relate to any of the other SPI products, feel free to drop me a line. Alternatively, you could look at our website. The address is there. I think on there there's actually a facility to ask any specific questions. So it just remains for me to thank everybody for taking the time out to listen to the webinar today. I know you're all busy people, and we really appreciate taking some time out of your day to share learnings with SPI. Wishing you all the best. Thanks.
Thank you so much, Graham, for that great discussion. That was very insightful. Really appreciate your time. And thank you all on the webinar for joining us today to discuss the many benefits of incorporating UltraBurst technology in the development of your next product. Let's now address the questions that have been asked during the webinar, and we'll jump into the Q&A. So to address the first question that was asked, the question was asked, when formulating with UltraBurst, what other excipients can you use and what level could you use them at?
Okay, so just to emphasize, UltraBurst has been designed as a ready-to-use platform. So in terms of other excipients, not required in general, other than lubricant. So we suggest to add either magnesium stearate or sodium beryl fumarate at around 1–1.5% level. Simply blend that with the API and you're good to go. And we have been, I think, as I said in the webinar, we've had drug loadings up to around 30% and higher, and been able to form robust tablets. So our general recommendation is just essentially take your API, whether it's Tastemaster or not, blend it with the UltraBurst platform, add some lubricant, blend that, and then you're good to go. And that will cover all APIs at all levels. So hopefully that answers that question, Andrew.
Yeah, it does. Thanks, Graham. So the next question that was asked, what is the best shape for ODTs? Standard, concave, or FFBE, flat-faced, beveled edge?
Okay, so most of our work has been done with a flat-faced beveled edge purely because in terms of friability, that's the most challenging shape. And so all the data we presented is generally with that shape tooling, and I think that's the most commonly used kind of shape for ODTs. If you look at a lot of the products on the market, they come in that shape. You don't have downstream coating processes for ODTs, so that shape is good for ODTs. That's not to say that other shapes cannot be used. Remember, we designed the UltraBurst system to produce robust, high tensile strength tablets, and therefore other shape tooling can work, as can the inclusion of a logo on the tablet, and that's also work we've done. We've seen really nice clear logos using UltraBurst. So really, there's not a limitation when it comes to tooling and the shape of the tablets. So hopefully that one's covered.
Yeah, that's perfect. So the next question that was asked, typically, what do UltraBurst tablets taste like?
Okay, what do they taste like? So this is one of the, I think, unique kind of attributes of UltraBurst. As mentioned, we have a formulation that has an effervescent type component to it. So typically, there's a little bit of a sensation of citrus or citricy type taste. So it really goes well. That really goes well with flavors such as orange, raspberry, etc. And actually, we have prepared. If anyone's interested, and they can contact us after the webinar. We have prepared some prototypes of UltraBurst tablets, so folks can try them out for themselves. And as I say, particularly with the kind of citricy fruit type flavors, maybe a little bit of sweetener as well, depending on your taste. We really get a very nice, refreshing organoleptic experience using UltraBurst.
Awesome. So the next question, do you have data on disintegration in a case where direct compression is not possible and roller compaction has to be applied before tableting?
Okay, so we have looked at, we've done a little bit of work on roller compaction using UltraBurst. We have been able to form fairly robust granules, and this is obviously good when it comes to matching particle size of the UltraBurst with, for example, a taste mask coated API. And when we use those granules, the resultant product had, I would say, there was a slight decrease in the DT, but not a really big decrease in DT. So again, I would say we probably haven't optimized the process around the hardness, etc., of the granules, but it's certainly something that we believe is doable, and we retain significant functionality of UltraBurst using that approach. So, but it's a really great question, and it's something that we have thought about and we have worked a little bit on. But there's probably a little bit of more work that we could do in that area. But thanks for that question.
Great. And so the next question, it was asked, have you found any need to add additional excipients to enhance tablet strength and friability? For example, addition of MCC as a binder?
Okay, so covered that a little bit earlier on, but we designed the system to try and optimize the combination of disintegration time with the high tablet strength. The system builds on our XL technology, where we have higher compactable mannitol. So typically we haven't seen the need for the addition of other excipients, and particularly MCC. We know that it can give a certain sort of grittiness or not so nice mouthfeel if it's added in too high a level. So thankfully, at the levels and at the APIs and the formulations we looked at, we've not had to add other materials, and I think in general we would caution against that, because it does change the organoleptic properties of the system. But that doesn't mean to say that in certain examples maybe it's worth a try. But I think we'd want to talk to folks around reasons why they would want to add other materials and whether or not we could we'd recommend that. But in general, we've not had the need, we've not seen the need to do that in the work that we've done.
Thanks for addressing that. We have time for one more question. It was asked, are there any negative interactions with excipients?
Okay, sorry, to cut up a little bit there, Andrew. I think the question was, is there any negative interactions with any APIs or other excipients?
Or other, or other excipients, exactly.
So again, we've worked with a range of different APIs, and I would say we haven't seen anything specific to the components of UltraBurst that are unique to UltraBurst in terms of interaction. We've had a couple of examples where there's been some issues, but that's been caused by, for example, addition of sucralose to the system, which was not anything to do with UltraBurst. It was more to do with, we found the root cause of that was the supply of sucralose and the conditions in which the tablets were stored. So we actually found that very dry conditions, there was formation of black spots, which we only picked up after a little while. But we signed that specifically to the manufacturer supply of sucralose. I think there's one other example of an interaction with mannitol, but that was a known interaction with mannitol, so not surprisingly, we have mannitol in the system, so we also had some issues there. But in general terms, it's been pretty good in terms of the overall compatibility with a range of different APIs and different excipients, flavors, and colors. Again, hopefully that answers that question.
Yeah, that's perfect. Thank you, Graham, for addressing all those questions. That was really appreciated. And if for the questions that Graham did not answer, he will reach out to you directly via email to help you address those additional questions. And thank you all, the attendees, for joining today and for your time. It's a pleasure. Hope you guys have a great day. Thanks again.